Okay, yeah, I think we can get started. Welcome everyone to this week's Connect Science seminar. I'm very excited and honored to introduce our speaker today, Dr. Howard Cedar, professor at the Hebrew University Department of Developmental Biology and Cancer Research. Dr. Cedar started with a BS in mathematics in MIT and received an MD and a PhD from NYU. He moved to Israel in 1973 and became a full professor in 1981. He is a world-renowned scientist in the field of DNA methylation and defining its influence on gene expression. He's been working on DNA methylation for over 5 decades and appreciating his pioneering discoveries, he's received many prestigious awards, including the Israel Prize in Biology, Wolf Prize in Medicine, the EMET Prize in Life Sciences, and Gairdner International Award, and many others. He's also a member of the Israel Academy of Sciences and the U.S. National Academy of Sciences. Today, we'll be hearing more about DNA methylation from the world expert. Before passing it off to Dr. Cedar, just one housekeeping point. Please type your questions not into the chat option, but into the Q&A option, and we can have Dr. Cedar answer them towards the end of the presentation. Dr. Cedar, thank you for being here today, and thank you for your time. Please take it forward.
Okay. Thank you for inviting me. It's a pleasure to be here. And I love to have the opportunity to tell people about DNA methylation, because there are a lot of basic aspects of DNA methylation that are not really well understood, even by scientists who work in the field. And I think that it's important to really understand the guts, the concept of DNA methylation, and I want to devote my my talk to that. There'll be I'm sure that everyone will see that what I'm going to say is really applicable to a wide range of different subjects and diseases and cancer and aging, but I'm not going to talk specifically about those things. Okay, so first thing is that I'd like to just mention the people who took part in this study, the main people who took part in the studies I'm going to tell you about. Tal and Sachi and Shari were students, and Ofer and Howie are technicians and lab managers. And some of the work I'm going to present was done in collaboration with Yehudit Bergman and Ben Berman at the Hebrew University and Ben Stanger from the University of Pennsylvania. So if you take a look at the distribution of DNA methylation in the genome, you see a picture that looks much like this. Almost the entire genome is methylated, not to 100, but almost to 100%. And then there are these wells over here with no methylation. And these are CPG islands. CPG islands are scattered throughout the genome. They're mostly promoters of housekeeping genes and as well as other kinds of genes, right? And they are unmethylated. And if you take a look in any cell of the body, this is the general picture that one sees. Methylation everywhere except for the islands. Okay? And this is true for all cells. How does this come about? Well, there's a methylation pattern that comes on the DNA from the sperm. There's a methylation pattern that comes from the DNA of the oocyte. In the early embryo, these methylation marks, are all erased, almost completely erased. In other words, the identity of the oocyte and the sperm through methylation is erased. And now the system starts from scratch. And the first thing that it does is So you see here are the DNA that comes from the male and the DNA that comes from the female. And you see everything is unmethylated. And then at about the time of implantation, there's a wave of de novo methylation that methylates everything. But CpG islands are protected, and they're protected by virtue of cis-acting sequences that are located within these CpG island regions. You see there are many CpGs here, but because of certain cis-acting sequences, they're protected. Now this pattern, this concept of de novo methylation and protection is only true at the time of implantation. But the pattern generated by this one-time thing, okay, is then preserved or maintained throughout the life of the organism. Throughout, even through replication, this pattern is maintained by autonomous mechanism of copying, okay? And it works like this. So if this is a piece of DNA, any piece of DNA, you'll note that CpG is a symmetrical dinucleotide. So if there's a CpG in the 5 to 3 direction in one strand, there's a CG in the 5 to 3 of the other strand. This is a site that's unmethylated. All methylated sites are methylated in both. And then during the time of replication, the two separate strands are replicated. In other words, the sequence is copied, and this generates a hemimethylated site. And the hemimethylated site is then recognized by DNMT1, a methylase, along with a number of other factors, which only know how to methylate if the DNA is hemimethylated. And these sites, which were totally unmethylated, will not get methylated as a result of this simple copying mechanism. The methylation pattern of the daughter cell is copied to the mother cell is copied to the daughter cells. As a result, and this, if we take a look at adults or even old people, okay, and we look at their methylation pattern in different tissues. So blue here is undermethylated, yellow is methylated. Here are a whole bunch of samples of brain DNA, liver DNA, muscle DNA, colon DNA from a number of different people. They're all about 70 years old. You see all the islands remain unmethylated as they were set up at the time of implantation. And all the other sequences remain completely methylated. In general, you'll see that there are fine details here that we are not seeing, but in general, the pattern is maintained through old age. So, and the pattern of methylation in our genome basically was established years ago, but maintained by a very potent maintenance process. And DNA methylation generally causes repression. It does this because when DNA methylation is on the DNA, it can dictate protein or chromatin structure. So in general, during the time of replication, the DNA is unmethylated and it gets replicated. So the DNA polymerase comes plowing through. It displaces the chromatin structure to some extent, okay? But if this DNA didn't have methylation, it'll get repackaged in a structure that's relatively open. As opposed to that, DNA that's methylated, here too, the chromatin basically gets erased by the movement of the DNA polymerase, but because this region is methylated, it generates a more closed structure. And in this way, the accessibility of genes is preserved through a long period of time by virtue of the fact that you have DNA methylation as a stabilizing influence. After implantation, the next stage is basically to turn off pluripotency. And there are a number of genes that are responsible for pluripotency, like OCT4 and NANOG and a bunch of other genes that are important, okay? And I just want to show you how these genes are turned off, okay? So here's an example of a region of the OCT4 gene. You see that the DNA itself is unmethylated in this control region, but it has a histone structure that's active, with the histones being acetylated and lysine 4 being methylated. Right? The first thing that happens in the inactivation of OCT4 is that there's a repressor, a protein repressor that recognizes the gene and turns off RNA synthesis. First step. Okay. The second step is the recruitment of the enzyme G9A, which is a histone methylase, and it recruits histone deacetylase, and these together bring about a removal of all the acetylation and removal of the methylation at lysine 4. Then G9A methylates lysine 9. This attracts heterochromatin protein. And now, in addition to the gene not being synthesized, now the RNA being off, now the gene has converted into heterochromatin. Then in the last step, the same G9A recruits the de novo methylase, and that methylates the DNA. So pay attention to the fact that it's not the DNA methylation that's regulating the activity of OCT4 or any of the pluripotent genes, right? Rather, the pluripotent genes are turned off, then heterochromatinized, and as a last step, they're methylated. But if you don't get the methylation, if you get the turn off of RNA, right, and you even get the heterochromatinization without the methylation, okay, this gene will go back to being on. It's not stable, but as soon as it becomes methylated, the inactivation becomes stable. And that's the role of DNA methylation, not to change the activity, but to stabilize it. Okay, the next step is the formation of ectoderm, mesoderm, endoderm, the germ layers. And then you get the organogenesis, where you form the different tissues. And here, we know what happens. We know that in each specific tissue, okay, during the process of differentiation, certain genes, and this occurs mostly at enhancers, undergo specific demethylation. Okay? So specific demethylation in the liver when it is getting developed, and specific demethylation in the colon during development. Again, okay? Note that this is specific demethylation. So if we now go back, we see in the early embryo, everything here, all these sites were basically unmethylated. At implantation, all of the DNA got methylated except for the CpG islands. The CpG islands remain unmethylated in all tissues, but these, many of these sites here, especially those in enhancer sequences, undergo specific demethylation in the tissue that is being developed. Okay? This is for the liver, for the colon, for muscle, for spleen. Each one has specific tissue-specific DNA methylation. I just wanted to show you that what I showed you before was just a diagrammatic picture, but here is a picture just published by the laboratory of Yuval Dor. It was published in Nature, which is an atlas of specific DNA methylation sites in over 40 different pure cell types. And you can see it has exactly the same concept, the same idea. Okay, how does this demethylation occur in tissue-specific manner? So if we look in B cells, for instance, we can find all of these genes, all of these regions that are specifically unmethylated in B cells. They are methylated in other tissues. They were methylated at the time of implantation. Okay, over here. Okay, and they undergo demethylation. The demethylation we have shown is carried out by the TET enzymes, TET2 and TET3. And if you do a knockout of TET2 and TET3 prior to the B cell stage, you prevent this demethylation. So this demethylation doesn't occur. You still get B cells, cells that look like B cells. Okay? But now, if you look at these cells, you'll see, now this is looking at the RNA. So in the wild type, these are many RNAs associated with those enhancers, which are active in normal B cells. As a result of the knockout, okay, many of these genes are not expressed or express less. So the demethylation is important for function. Without the demethylation, you don't get stable function, or you don't set stable expression. Okay? And it's also important for function. So if you look at the formation of germinal centers in response to an antigen, the wild type gives a normal number, but the TET2, TET3 double knockout and is greatly impaired in making germinal centers. So from the point of view of expression and function, DNA methylation, demethylation is necessary to get a stable cell type. Okay, and of course, once this occurs, once you've gotten this demethylation, okay, because of the maintenance mechanism, that pattern will remain for the rest of the lifetime or even after replication of the differentiated cell type. It'll remain unmethylated. Okay. What about if you look at these enhancers? You see that in the normal B cells, in the wild type, these enhancers are accessible. In other words, they're positive for ATAC. Okay? But in the knockout, you don't get the accessibility. Okay? Furthermore, if you look at specific transcription factors that bind to these enhancers, you see the wild type binds these transcription factors, but in the knockout, this is greatly impaired. Again, demonstrating that what I said before is true, that basically methylation works through chromatin structure. That's how it affects gene activity, through chromatin structure. Okay, so if we summarize this part, you can see that this is a gene, this is the promoter. Many times these promoters are CPG islands, and they're anyhow unmethylated, but the enhancers are methylated, okay? and they're methylated in all tissues. And then in the specific tissue, there are specific factors that recognize these enhancers despite the methylation and their closed chromatin structure. They're recognized. These recruit the demethylase. The demethylase causes demethylation. That releases the chromatin structure. So now the structure is open. Okay. And this allows the binding of other factors to these enhancers, which then affect the expression of the gene. And I must emphasize the fact that many of the factors that bind to these enhancers are not specific. They're general factors that are present in almost all cells. And they're the ones that really activate the promoters. Right? So what's controlling this system is the demethylation. If you don't get the demethylation, you won't be able to get the enhancer activity. Okay. So it turns out that changes in DNA methylation not only are associated with development, but can also occur in the adult or postnatally. So here's an example. If you take a look at the sites that, again, enhancer sites, there are a lot of sites that are found to be in the liver unmethylated in the male, yet methylated in the female. And when you ask, how did this come about? When you look at the time of birth, all of these sites are methylated. And then as time goes on, and this is 3 weeks, 8 weeks, you see at 8 weeks already, right after 3 weeks, 8 weeks, the male starts undergoing demethylation at these sites. And then in the adult male, it's completely demethylated. So here's a process that occurs postnatally, and it occurs only in the male. Okay. Now, how does this happen? Well, it turns out it happens because in the male, at about 3 week time, he starts secreting testosterone. And it's the testosterone that mediates this change in methylation. So here we have a male, for instance, okay, who is undermethylated in the liver. If we castrate the male early on, okay, he won't demethylate those sites. In other words, it requires testosterone to get the demethylation. But if you castrate them and add back testosterone early on, he'll still demethylate. So it's necessary and sufficient for the demethylation process. And you can even take a female and early on give her testosterone, and she will also bring about demethylation. So here is a process that occurs postnatally, and keep in mind it's mediated by hormones. Okay. The fact that it's mediated by hormones, right, can also be seen here, for instance. This is a different tissue. So in the hippocampus, there are also sites that are unmethylated in the male, but methylated in the female. Okay. And here too, this is controlled by the testosterone. And the same thing is true for fat cells. And these are sites different than the ones we saw in the liver that are undermethylated in the male, but methylated in the female. And by castration, this does not occur. So again, all of this, and this is in humans, by the way. Don't think that humans are different. That's for mice, but in humans, it's exactly the same thing. Okay. Postnatal demethylation occurs in every tissue. And here's an example of the liver. So what you're looking at here are sites, okay, that are specifically unmethylated in an adult liver. Okay. Other tissues, they're completely methylated, so it's specific. But if you look at the time of birth, you see that there are some genes, some enhancers, okay, that are methylated. In other words, this occurred during development. All this occurred during development, and at birth they remain demethylated. But these sites never underwent demethylation during development. Right? And then, as you go on, you see that after 3 weeks, at 3 weeks, many of these genes undergo demethylation, okay? But these remain methylated. And only later on do these sites become demethylated, right? And I just want to take the time to show you what happens if you knock out the ability to methylate, the mechanical ability to methylate by TET, okay? So for instance, if you knock out the ability to demethylate at birth, okay, then the pattern that exists at birth remains even in the adult animal. In other words, this is not a dynamic process. This is a process that happens once by TET and then gets maintained. It's stable, right? If you knock out at 3 weeks, right, the pattern that was at 3 weeks remains. You don't get remethylation or any other demethylation. After that, it's stable. I'd just like to point out another aspect of this. So here are those sites that undergo demethylation during postnatally. I put them all together here. Some of them then undergo demethylation by 3 weeks, and then the rest of them undergo demethylation after 3 weeks. Okay. Note, okay, that if you make a knockout, a postnatal knockout of the insulin receptor in the liver, postnatal knockout, okay, this demethylation does not occur, indicating that this process, all of this process, is again controlled by hormones. Okay. We think that this demethylation, okay, has to do with nursing. In other words, until birth, the embryo is getting its nutrition from the mother, right? And then, following birth, the animals start drinking milk, which is rich in fat. And we think that these demethylations represent an adaptation to nursing. While, again, after 3 weeks, these animals are weaned, and that the weaning again changes the form of the diet, and this brings about an adaptation mediated by insulin to the new diet. If you take a look at the liver in pregnant females, okay, you have a pregnant female as opposed to a virgin. You'll see that all of these sites are methylated in the virgin, but in the pregnant liver, in the liver of the pregnant female, all of these sites undergo demethylation. This is hormone mediated and clearly an adaptation to being pregnant. The liver undergoes an adaptation to the state of pregnancy. And this site, once it occurs, remains, so that in females that were once pregnant and are no longer pregnant, their liver is already primed for this adaptation. Not only that, but it is probably this that allows, that controls the fact that the liver can regenerate much better in pregnant animals. Okay. Now I want to show you a case of change in methylation that has more to do with environment. Okay, up to now, we've been looking at cases that are indeed induced within the organism through hormones, right? But they're not necessarily adaptations to what happens in the environment. So here's a case. I'm showing you here how muscles are formed. Okay. So there are satellite stem cells in the muscle. These get activated. They form myoblasts and myotubes, and then make up a mature muscle as they differentiate. These cells can proliferate, okay, and then differentiate. So now, if you take a look at muscle stem cells, these satellite cells, and you injure the muscle, you physically injure the muscle. As a result, if you now take muscle stem cells from those mice that were injured, okay, all of these sites have undergone demethylation. In other words, the muscle stem cells have undergone some form of activation. Okay. This activated state is a memory, so that for another months and months and months after that, and maybe longer, the satellite cells in these mice, in this muscle, is no longer the same satellite cell. They're activated. And this remains with the animal. Okay? This is an adaptation to the injury. Not only that, if you now take a look at other muscles in the body that were not injured, they also undergo this same activation. One injury brings about activation of all the muscles in the body. And as you would guess, this is mediated by hormones, the hepatic growth factor. And this, we know with studies I'm not going to show you, that it's this undermethylated state which allows muscles anywhere in the body to respond quicker to a subsequent injury. Any other injury later on, the muscles will respond and regenerate much quicker because of this demethylation, which remains stable for the life of the organism. Interestingly, in pregnant animals, the muscles also undergo this activation to some extent. Okay? Probably, again, the pregnancy through hormones induces this activation, this readiness, adaptation in the muscle cells. So if we take a look at how this process works, we see something like this. Okay. Hormones that are induced by events, either internal events or external events, can interact with cells. Okay. this brings then through signal transduction, this brings about the induction of transcription factors. What you see here is an enhancer region, okay? The transcription factor binds to these enhancer. These are like master transcription factors. They can bind to these genes. They recruit a demethylase, okay? And that they go undergo demethylation, okay? And once they've undergone demethylation, they can recruit acetylase and the lysine 4-methylase and become open, okay? So they're now unmethylated and open, okay? And this will allow other transcription factors, even general transcription factors, to come and influence the expression of the gene. Okay. Now, you would think that if we take a look at the methylation patterns in all the tissues of the body, and then ask in a non-biased manner, how are all these methylation patterns related to each other? Okay. So these are all differentiated tissues, all of these. Okay. And if you do an undirected analysis, an unbiased analysis, it always comes out like this. That is that the mesoderm tissues are connected, the ectoderm tissues are connected, the endoderm tissues are all related, and a fourth category of blood cells are also all related. Okay? Strongly suggesting that there are changes in methylation that occur early in development that get carried on. And so that they're present in all the different tissues. This would explain how this happens. But this is not the case. So for instance here, if you take a look at the embryo, early embryo, this is a 7.5 day embryo, and you isolate the cells from ectoderm, mesoderm, or endoderm, and look at the methylation pattern, you see that many sites that got methylated at the time of implantation undergo specific demethylation in ectoderm, mesoderm, endoderm, okay? But if you now look in adult tissues, these sites have become remethylated. So there is specific demethylation in these tissues, okay? But then this gets remethylated. If you take a look at the sites and ask what factors interact with these sites, okay, all of them interact with very highly specific and related factors. So factors that govern ectoderm bind to these sites. Factors that are important in endoderm bind to these sites. And factors that are important in mesodermal lineages, in the early lineage, bind to these sites. Right? So it looks like what's happening is that by demethylation, you turn on activities in these cells. Let's say, take the endoderm, you turn on activities in the endoderm that are needed for the primitive and for the early stage of the germ layer. But once the germ layer has started further differentiating, these are turned off, not needed anymore, and they're turned off. So here is an example of something that we've never seen before. That is demethylation and subsequent remethylation. Okay. Much different than the model that I've been showing to you up till now. Okay. Later on in development, at about 10 or 11 days, there are new sites that undergo specific demethylation in ectoderm, in endoderm, in mesoderm, and these, by and large, are maintained in adult tissues, thus giving rise to this dendrogram, okay, which shows all of the mesoderm together, all of the ectoderm together, all of the blood together, and all of the endoderm together. And these are embryonic tissues, which also are different in all of them, right? And migrate differently. Okay. I want to go back now for a second to the case of the muscle injury. Okay. So I want you to take a look now at the following picture. An implantation, these sites are methylated. In muscle stem cells, these sites are methylated. Okay. And upon activation, they become demethylated and remain that way in adult muscle. In all other tissues, these are methylated. They're tissue specific. But there are also sites, many of them, that are specifically demethylated in muscle stem cells. But look what happens when you activate them. They get remethylated. So they originally got demethylated for the muscle stem cell. Now they get reactivated. Again, these are sites that are needed for the stem cell state, for the important things about being a stem cell, and then get remethylated with the onset of differentiation. Completely different model. And this is true for stem cells in general. Here I want to show you that all these sites are specifically undermethylated. They're specifically undermethylated in hematopoietic stem cells. All of these are specific. This is what you see in other tissues. They're all specific for hematopoietic stem cells. When these hematopoietic stem cells differentiate into all the various blood lineages and branches, all of these sites remain unmethylated. But there's a whole category of sites that become remethylated. And these are marked. Okay. I'm going to show you now. If you now look in these stem cells, okay, and take the sites. So you saw before there are two types of sites. There are sites that remain demethylated, and there are sites that become remethylated. But in the stem cell, they're all unmethylated. You now take those sites and you look in the stem cell at the presence of, for the presence of lysine K9 methylation, okay? Lysine K9 methylation, you find the following. The sites that remain unmethylated, okay, are not marked. The sites that become methylated are already marked with lysine 9 methylation before they become methylated. So somehow this is serving as a mark, okay, for which sites will become remethylated in this system. And what I'd like to do is finish up just by giving you an overall picture of what I just told you about methylation, because we now have a situation, we now understand that methylation is not changes in methylation are not inherited forever. They can be changed. Okay? And what these studies have suggested is that there are two types of paradigms. There are many enhancers that are marked. They have methylation, okay? And they have a site for the binding of a specific factor, okay, which can bring about demethylation. Okay. Now, these sites have no facility ever to become remethylated because they don't have the programming for it. There's nothing there that will allow the remethylation. There's no more generalized methylation anymore. Only if specific methylation can occur. And if there's no instructions here, it won't occur. In contrast, there are some sites that are programmed for this. So they have a site for bringing about demethylation, right, through TET, but they also have a site that can bring about the remethylation through recruitment of de novo DNA methylases, okay? And I think this is probably important because it gives us now a clue on how we may be able to go back in development and regenerate methylation patterns which will allow even stable tissues to go back and regenerate. And with that, I'd like to end my talk and thank you very, very much for your attention.
Thank you so much, Dr. Cedar. That was really very clear, and I think everyone that had attended this presentation would have gotten a fundamental understanding of how DNA methylation works. There are a few questions on the chat. I would like to start reading them out to you. Printed. Excuse me. Can you hear me?
Yeah.
If you can hear me, can you hear me?
Yeah, I can hear you.
Sorry, I'm losing audio. You guys, can you hear me now? Sorry.
Yes. Yes.
Okay, sorry. How about imprinted genes? Example, H19 IGF2 loci. Do they also undergo demethylation and de novo methylation? And wouldn't that erase the memory of imprinting? Which allele came from father versus mother?
Yeah, that's an excellent question. And it turns out that the imprinted genes, okay, come from the gametes with their own methylation pattern. Okay, but on the basis of what we've been discussing previously, in the early embryo, all of these sites would be erased. But it turns out that imprinted genes are programmed for a unique process in which it uses histone methylation as an aid in preserving the DNA methylation pattern, the original parental DNA methylation pattern. Okay? Again, it has to be programmed. If it's not programmed, it won't work.
So also another follow-up was is G9 from the same person is how G9A HDAC recruitment leads to H3K4 demethylation.
So I didn't put in all the details, but K4 methylation demethylation is brought about by the recruitment of another enzyme, not the HDAC. Yeah.
I see. Another person is asking, what's the role of methylation quantitative trait loci, MQTLs, in all these procedures? MQTLs are involved in the recruitment of TET enzymes and induce tissue-specific demethylation. That's a question.
Yeah. So, as you saw here, all of the action of methylation occurs at enhancers or regulators. Okay, there are also silencers or enhancers. Okay, and so changes in many. What happens is that many polymorphisms. Okay, actually bring about a change in methylation. And it's the change in methylation which brings about the different expression patterns. Okay, so DNA methylation at these sites plays a big role. And you know that many investigators have pointed out that even for diseases that we know that there's in a sense genetic aspect to it, it only accounts for a small number of the cases, a very small number. And one of the ideas is that because it brings about the differences through DNA methylation, which are not being observed in these studies.
Right. Yeah, that's great. Dr. Ramesh Shivdasani is asking, do the TF motifs enriched in remethylated enhancers provide a clue as to which transcription factors may be responsible? If so, are those transcription factors the same or different in, let's say, blood versus liver?
Yeah, so they definitely provide a clue, okay? Definitely provide a clue. And yes, probably they're different in different tissues. Absolutely, yeah.
Mm-hmm.
So
and
you realize that all of these enhancers are really tissue specific, so that the same enhancer is remaining methylated in the other tissue, right? And the major reason is that because that other tissue has different pioneer factors. Right. So it's basically all determined by that.
Yeah. And on that note, we have cellular reprogramming in advanced, certain solid tumors and also in other cancers. So in that case, do you think these enhancer demethylation is pretty stable, or are they plastic in nature where they can go back and forth in terms of demethylation?
It's a great question, and we really want to work on this. I think the basic question has still not been addressed in an organized manner, and that is to see whether the methylation pattern of tumor cells, right, actually includes an epigenetic de-differentiation to earlier cell types. And we would love to do that. I think it's a very, very important question.
Mm-hmm. And how broad are these methylation sites supposed to be? Are they a cluster of, let's say, 10, 15 sites to CPGs together, or even one specific site can make a difference? Can you comment on that?
Yeah, so these usually cover a region of somewhere between about 400 base pairs, right? Between 300 and 400 base pairs, right? And they usually include something like 2% CPG. Okay, so they could have 8 sites. They're not islands which have many CPGs, but they have sites, and all of the sites change. Exactly, it's a regional. I wouldn't call it regional. It's not big, but it's definitely a local phenomenon.
Mhm. Another broader question is, can you comment on how these systems may be involved in observed inheritance patterns related to famine survival?
Yeah, so this is a whole other subject which would take another 2 lectures to talk about.
Mhm.
But yeah, so it's a major question, and that is this so-called transgenerational inheritance, where the behavior of the parent can influence the behavior of the offspring. Okay, and even though we don't know this has never been proven, I think that, at least from our work, we can say that the transmission from the gamete, the transmission of the information, the experience from the gamete, is mediated by hormones. It affects the gamete. The information passes through to the early embryo, probably by RNA. Okay? But I would bet, and we have some evidence for this, that it is then remembered in the offspring by DNA methylation. Again, you must distinguish between activity and memory and stability. Yeah.
Mm-hmm.
So here too, I believe that there's a role of methylation. It hasn't been 100% proved yet.
Another question regarding whether there's any sort of like a correlation between hormones in modulating DNA methylation and SAM levels in these tissues.
Yeah, this is a whole big subject about folic acid and SAM. And yes, I think that this has a big influence. Absolutely. And in fact, this has been shown. There are studies on this. Yeah, for sure.
Another broader question is, can you please comment on whether demethylation and remethylation will cause chromosomal instability in elderly?
I don't know. I really don't know. But I can tell you that a completely different type of methylation that we haven't spoken about here, that occurs over a lifetime, is an indicator of aging, as many of you know. Okay. And to a large extent, I have a feeling that it's the methylation process itself that affects the aging. But here again, this is a big challenge.
That's great. Another person is wondering, in terms of your insulin receptor knockout and how that induces postnatal demethylation, if you can comment on how obesity affects postnatal demethylation.
Yeah, we don't know. It's certainly possible. We haven't looked at this. Okay. We actually did look at it, but it turned out we looked at it the wrong way. So I don't have any information, but certainly that might be the case. Yeah, I don't know. Considering, the thing is that all of these things are mediated by hormones. So anything, I think, maybe not anything, but a lot of things that cause changes in hormones may in the end have an effect on this DNA methylation in a stable manner in the organism. Yeah.
All right. Thank you very much, Dr. Sita. Those are all the questions, and I think it was a fantastic talk. And on behalf of the Cancer Connect Science community, I would like to thank you for taking the time to present and look forward to more exciting research and breakthroughs coming out of your lab. Good luck.
My pleasure. Thank you. Bye-bye.
Bye-bye.